In this video, I will describe how chemical signals, aka neurotransmitters, generate electrical signals in neurons. We know that the nervous system is made of billions of cells. The functional cell for the nervous system, the neuron, is made up of a cell body or soma, where dendrites branch off from, and an axon, also attached to the cell body, which eventually forms axon terminals that release neurotransmitters. Neurons come together to form circuits in our nervous system, and the way they come together or talk to each other is through synapses. If we think of the simplest neural circuit involving two neurons, we will see that we have both a presynaptic neuron, i.e. the neuron before the synapse, and a postsynaptic neuron, i.e. the neuron after the synapse, that interact via a single and sometimes multiple synapses. A synapse is essentially any connection between an axon terminal of a presynaptic neuron and the dendrites or the cell body of the postsynaptic neuron. In this two-neuron circuit, the presynaptic neuron will release a neurotransmitter through its axon terminals onto the dendrites or cell body of the postsynaptic neuron. It is at the dendrites of the cell body of neurons where there are receptors that respond or are activated by neurotransmitters released into the synapse. If we consider the types of neurotransmitters released from presynaptic neurons onto postsynaptic neurons, there are three main types or classes. Specifically, neurotransmitters can be excitatory, inhibitory, or neuromodulatory. Of course, excitatory neurotransmitters will act to depolarize neurons, or increase the probability that they will fire off an action potential. On the other hand, inhibitory neurotransmitters will act to hyperpolarize neurons, or decrease the probability that they'll fire off an action potential. Neuromodulators, which we won't discuss in this video, tend to act more slowly and will bias neurons towards being excited or inhibited. But how does this work? An important point to note is that neurotransmitters are not necessarily excitatory or inhibitory. It all comes down to the receptors that they bind to. Let's zoom in on an excitatory synapse. An example of a neurotransmitter that depolarizes neurons and causes excitation is glutamate. Glutamate binds to postsynaptic receptors that open sodium channels. So whenever glutamate is released, it causes sodium influx, which makes the membrane potential of the neuron more positive and more likely to reach the threshold for an action potential. Let's consider that there is another presynaptic neuron acting on the same postsynaptic neuron, but this time it is an inhibitory neuron. Let's zoom in on one inhibitory synapse. An example of a neurotransmitter that hyperpolarizes neurons and causes inhibition is gamma-aminobutyric acid, or GABA for short. GABA binds to postsynaptic receptors that open chloride channels. So whenever GABA is released, it causes chloride influx, which makes the membrane potential of the neuron more negative and less likely to reach the threshold for an action potential. Glutamate and GABA both act on ligand-gated ion channels, or channels that are deemed chemically gated, and these shouldn't be confused with voltage-gated ion channels, which generate the action potential. In neuroscience, we call the depolarization or hyperpolarization at the dendrites and soma of neurons postsynaptic potentials or graded potentials. Postsynaptic potentials and graded potentials essentially mean the same thing. When there is depolarization, this is labeled as an excitatory postsynaptic potential, or an EPSP for short. When there is hyperpolarization, this is labeled as an inhibitory postsynaptic potential, or an IPSP for short. Postsynaptic potentials should not be confused with action potentials. Action potentials are caused by postsynaptic potentials. Postsynaptic potentials can vary in their size and shape, but action potentials are the same size and shape. To understand the difference between postsynaptic potentials and action potentials, let's think about the structure of a neuron. Because we need synaptic input from the presynaptic neuron for postsynaptic potentials to occur, and synapses from the presynaptic neuron are located at the dendrites and cell body of the postsynaptic neuron, i.e. where the connection points between neurons are, postsynaptic potentials also occur at the dendrites and cell body. If we were recording the membrane potential at parts of the dendrites and cell body of the postsynaptic neuron, we would see small changes in the membrane potential wherever and whenever a neurotransmitter was released from presynaptic neurons, aka we would see postsynaptic potentials. To cause an action potential, postsynaptic potentials are summed or added up. Summation in maths is adding numbers together. This is essentially what happens at the dendrites and cell body of neurons. If there are lots of EPSPs, they will all add together and push the membrane potential above the threshold needed for an action potential. If there are EPSPs, as well as IPSPs, summation still occurs, but the IPSPs have an opposite effect to EPSPs, as they act to hyperpolarize neurons, or make the membrane potential more negative and push it further away from the threshold for an action potential. Depending on how much inhibitory input there is, IPSPs could prevent the neuron from firing. Put simply, IPSPs act to block the effects of EPSPs. Overall, whether a postsynaptic neuron fires an action potential is dictated by the excitatory and inhibitory sum of the presynaptic neurons that synapse with it, whereby there can sometimes be thousands of synapses for a given neuron, allowing the nervous system to exert a precise control over individual neurons. Postsynaptic potentials can also be summed in space and time. Spatial summation is when there are many presynaptic inputs activating the postsynaptic neuron at the same time. Temporal summation is when a single presynaptic input is activating the postsynaptic neuron multiple times in quick succession. But Jacob, how do postsynaptic potentials cause action potentials? So, the summed or net potential caused by excitatory and inhibitory inputs travels towards the axon. At the axon hillock, which is where the cell body connects with the axon, is where the action potential is initiated. If the sum potential is enough to reach the threshold for an action potential, voltage-gated channels will open at the axon, and this will generate an all-or-nothing depolarization, aka an action potential, which will propagate down the axon. This highlights another difference between postsynaptic potentials and action potentials. Postsynaptic potentials are at the dendrites and cell body of a neuron, and action potentials are at the axon. If the summed potential is not enough to reach the threshold for an action potential, there will be no action potential. Keep in mind, when there is an action potential in the postsynaptic cell, this will cause the eventual release of its own neurotransmitter, and then it will become the presynaptic cell for the neurons that it synapses with. Thanks for watching.